the majority of neurons in the brain, particularly in cortex, are not sending signals most of the time. So roughly a sort of classic sort of 80-20 rule that 20% of the neurons are sending about 80% of the spike. What these other neurons are doing and what they're for is something of a deep mystery. We know about what 15% of the neurons in the visual cortex are doing. There's the rest of it, which is the bulk of it. We have no idea what it's doing.
Mark Humphries is a computational neuroscientist, publishing over 50 academic papers looking directly at the activity of neurons in almost every region of the brain. He works on the forefront of a new field, Neuro AI, and builds computer models to help us understand the brain's deepest mysteries. He's also the author of the book The Spike, an accessible story about how our 86 billion neurons wire together to make us who we are.
You've dedicated a number of decades of your life to studying this thing that we call the spike, the neuron spike, kind of the communication language of the brain. Talk about the spike. What is it, and why are you so interested in it?
Yeah, thanks, Evan. Thanks for having me on. So you're right. So I have dedicated pretty much my entire scientific career to looking at the spike since my PhD days. And the reason we're so interested in the spike is because, as you sort of alluded to in your question, it is the fundamental way that the neurons in the brain talk to each other. So if anyone has done any sort of biology to a high level at school, they'll have heard of this thing called the action potential, which is the pulse of electricity that gets shot down the axon of a neuron that reaches the end, and it's a thing that lets a neuron dump neurotransmitter out this end of its axon and communicates to other neurons. And neuroscientists, by shorthand, we call this thing the spike, because if you see these things on an oscilloscope or on a computer screen now, you can see it's an extremely rapid jump and crash of voltage. It looks like a straight line, essentially, when you see it, because it happens so quickly. It's about 2 milliseconds it lasts for. So these little spikes of electricity are how the brain makes things in the world happen. So they're in to make everything happen. So I mean, the trivial example of me talking to you now, so the fact that my jaw is moving up and down, we know is driven by spikes, because we can know that there are muscles that are moving my jaw up and down. It's the contraction of the muscles. The muscles are contracting because they are receiving spikes from motor neurons in the across the spinal cord and across the brainstem that are driving spikes, creating a release of acetylcholine onto the muscles. The tissue is then contracting under that release of the acetylcholine. And we know in turn that those spikes in the motor neurons are being driven by spikes that are coming from other neurons around them in the spinal cord, are coming from neurons that are in the brainstem or up in the cortex. And this whole cascade of spikes is driving the coordinated movement of the jaw and the lips and the tongue that's generating these sounds. And other spikes that we believe are generating then the words that are being spoken. So everything that's happening in the moment to you, to everyone else who's listening, is all about the spikes in your brain. So the understanding of what I'm saying, to see what you're seeing, everything seems to be driven by spikes. Hence, at a fundamental level, this deep interest in what the spike is and how it works.
How is it that one particular phenomenon in the not just the brain but also the body can generalize to all of these different modalities? Like neuron spikes give us vision, they give us consciousness in ways that we don't know, they give us art, they give us sport. How is it that one singular sort of physiological interaction can give rise to so many different phenomena?
So I'm going to say we don't know the answer to that question. So for, if you like, simpler functions, we have a fairly good handle on sort of the first parts of the process of how spikes do it. So we know a lot about, so I've just described to you a lot about how spikes from neurons can control muscles and therefore create movement. And we know probably more about that than anything. We're quite secure in the fact that we can work our way back from the muscle to the neuron, to the neurons behind it, and we can just follow the path of spikes backwards to go. So we can always electrically stimulate in, say, motor cortex, and we can make an arm move reliably in a particular direction because we know the pathway from those neurons in motor cortex, make them create spikes. Those spikes go down into the brainstem, the spinal cord. They cause a particular set of muscle contractions, and the arm will move in a fairly repeatable way. So that's a pretty solid evidence that these spikes coming out of here mean move your arm over there. But you're right that it is exactly the same spikes coming in, generating exactly the same way that end up creating our sense of vision in the world, our sense of hearing. So, and there we similarly can start from, say, the eye and the retina, where the light falls onto the back of the retina, and we know how photons are turned into spikes that get sent from the eyeball back into the brain. We know where they go, so we know they go to the thalamus, and after that they go to the back of the visual cortex at the back here, and we can trace them, and we can fairly well link those spikes to what they're responding to in the world. So, for example, if we recall from the very first set of neurons in visual cortex that get input from the eye, then we know there that those neurons tend to respond to tiny little pixels of the world. So each one is just looking at a different little bit of the world. And in that little bit, a lot of them tend to prefer an angle at it's an edge at a particular angle, shall I say. So, for example, some of them might like an edge at 45 degrees, some of them at 127, some of them at 90, so on. So the spike for one of those neurons means, there's an edge in this part of the world that is at that angle. And so we can follow that to the next area along and see that when those spikes arrive at the next bit of visual cortex, those neurons tend to pull together inputs from multiple of those initial neurons. So they kind of pull together different edges and say, these edges respond when these edges form a kind of line of some sort of corner. But as we get further and further into the brain, just like with the auditory system or the touch system, the further we get in, the more harder it is to understand what those neurons are now responding to. So from the sensory point of view, these spikes are ultimately ways of transducing external information in whatever form it comes to into some kind of common currency that can be used inside the brain. But the further we get from the outside world, the harder it gets for us it is to say what it actually means when it gets there. And that's just for the really simple stuff. But in your question, you alluded to things like consciousness and art. And that opens up an entire other can of worms, which is the problem that for many of the things that we like to know about how humans, the sort of unique cognitive abilities of humans, not just seeing and hearing, obviously lots of things do that, but things like language, things like maths, things like art, things like poetry, then we have literally no idea how spikes generate those things.
Yeah, that's going to be I think the hallmarker for this podcast overall is we just have no idea, which is brilliant because I think neuroscientists are so humble with their information. They're so great at saying we just don't know. That we know a few things, but we don't know a lot as well. And I think that we don't know thing should be echoed more in society today because there's far too many overly confident people making claims on things that we just simply do not know. But I think vision is a great example because I think you can understand how neurons sort of aggregate and summate information with vision. You get one neuron that does a horizontal line that activates in response to a horizontal line. You've one that activates in response to a vertical line, and you can start to see from the building blocks, like Lego building blocks, how you start to build this really complex picture basically out of lines, edges, colors, things like that. And that creates sort of our perception of vision, which is mostly an illusion. Are there any of those processes that we just either are a complete enigma? I mean, you mentioned consciousness. Like, what are the really interesting examples of those neurons that are taking quantas of information, like a line or an edge or a color, and building up this picture, but we just don't know how it's fundamentally doing it. And we don't know what the quanta or what the tiny bit of information is. Do you understand what I mean?
Yeah, let me see. So, because there's two ways to answer. So one way is that there are clearly processes where we feel like we know the beginning well, but very much so it falls apart further down. Uh-huh. So an example of that for humans would be, for example, reading, right? So given, as you say, a pretty good knowledge in the visual system of how edges at a particular angle come together to perform particular shapes, we can extrapolate to how the recognition of individual letters can come about, right? It's just another shape. We can just put together shapes. But the process from getting from that shape recognition to something which has a token that you understand as a separate entity. So, someone's name, some individual words, and then attach meaning to that, is way beyond our understanding. So in that sense, it's not clear how we would even know whether a set of spikes sent by a given set of neurons would stand for the understanding of the meaning of the word coffee.
Yeah.
What would that would even look like?
Yeah.
So, I mean, if we do look more crudely, we look in an fMRI machine, look at a whole brain scan, and look at its the blood flow in that brain, which should be proportional to the activity of the neurons. And we show someone a picture of a cup of coffee, a picture of a cup of tea, then there'll be a slightly different brain activation to the two, which implies there is something that stands for, this is the concept of a cup of coffee, a cup of tea. Even though visually they may look identical, you just tell them it's a different thing. But still, we have no idea. We get down to the level of the actual neurons, what they're sending to each other, what that would look like.
Yeah. To what extent, since all of these neurons seem like generalized processors of information, and they're phenomenal, as we said, in their ability to generalize information, to generalize stimulus, to what extent is that programmed by the environment, and to what extent are those neurons programmed by genes? And how does that interaction work?
Right. Yeah, there's a lot of good work on that. So there is so obviously the genetic component drives a lot of the scaffolding of how the brain is structured. Mm-hmm. So the clear sort of demarcated areas that we have in our brain, so that it's very clear that the genetics defines we've got this cortex with 6 layers, that we have a big massive cauliflower thing on the back, which is the cerebellum. Underneath cortex, you've got things like the hippocampus, which is to do with processing space and memory. And so those are all defined genetically. And it appears that quite a lot about sort of the different types of neurons that you find in different regions is defined genetically. So, for example, in cortex, in a set of the layers, you find this cell type called the pyramidal cell everywhere, which has a sort of little triangular shaped body, has a very long dendrite sticks out at the top, a little one sticks out at the bottom. And you can recognize them in all sorts of brain tissue from different animals. So they seem to be defined genetically, that they are a cell type that is specified, and it appears in a particular layer and not in another layer of cortex. That's all genetically defined. And at some point, it blurs into where development takes over. So the genes, famously, the mammalian brain, and particularly the human brain, obviously has far, far more cells than it can possibly be specified by a genetic program. So we can't have a genetic program that says there will be, all 20 billion cortical cells in a human brain. But each one of them will be a particular type of a particular position, a particular wiring. It just says, I'm going to have a whole bunch. We're going to have like 15 million pyramidal cells. We're going to have 1 million GABAergic interneurons. We're going to have 2 billion of these, and they're roughly going to appear in these layers. And they're going to be, some of them are going to send axons out of cortex, some are going to send it in cortex. So the scaffolding. So at some point, then development takes over and starts. So when the actual neuron ends, so for example, when a cortical neuron ends up in its position in cortex and starts growing out its dendrites and axon, then see the way it grows is specified genetically, but what it exactly connects to and quite how big it grows, that's all driven by development. It's driven by the sort of, if you like, the randomness of the environment that cell happens to find itself in, sort of the chemical environment around it, the genes it happens to be expressing at the time, because obviously a given gene can be expressed or not, but the amount of this expressed is quite stochastic, and the available building materials inside the cell it's not under its control. So if you might want to grow a really big dendrite, but it hasn't generated much protein to do it, it can't. So there's a lot of stochasticity in development. I should give a shout here to Kevin Mitchell's book Innate, which is fabulous on this stuff. But then that then shades into that sort of the development of in the womb, if you like, in the gestational period. And then as soon as you get to then that shades into the developmental experience of as soon as we emerge into the world, then there's a whole lot of stuff about our brain, which is about the neurons and the spikes they send, which is defined by the environment in which we find ourselves.
Yeah.
So there's classic experiments of what happens if you raise animals in an purely environment where they can only see vertical lines. Then it turns out in visual cortex, they don't develop neurons that can see horizontal. So you expose them to a world with horizontal lines in it, and they start wobbling because their visual system, and hence their balance system, has never seen them. They don't know what it is.
Bizarre.
So we know that there is even in that sort of fundamental level of sort of early visual processing, that's quite a little bit ends up being driven by whatever you're exposed to. We know, for example, that neurons tend to shrink and grow according to how active they are. So, which is generally a reflection of how much input they're getting from the outside world or they're not getting from the outside world. So ones are getting too much input, shrink down, try and get less, and ones are getting too much input, grow and try and grab more. So you can imagine then that if there's no horizontal lines at all, then if there's a whole bunch of neurons who are shrinking down or growing bigger because there's too much information of one kind and not from another, you end up with a miswired brain.
Yeah. So evolution has clearly packaged optimal development in, like, there's a large environmental stimulus is basically required to develop properly as a human, to have a properly balanced sensory system and to have a correctly wired brain, whatever correctly means.
Yes, exactly. So as you sort of touched on earlier, so the idea is obviously neurons are this generic processing unit. So you can do experiments. People have done experiments through the 90s, early 2000s, where you take a bit of cortical tissue from one region as just after an animal is born and put it, say, take the auditory cortex, put it in visual cortex, take some sensory cortex, put it in visual cortex, and find out what it turns into. And lo and behold, it turns into visual cortex. So it's not marked as some kind of special area of a cortex. It's just responding to whatever information it gets. And because it happens to be, by the genetic specification, wired, that part of the cortex is wired to the eyeballs, then that's going to turn into visual cortex. And because the bit over here is wired to the ear, that turns into auditory cortex, and so on.
Yeah, it's truly remarkable. So it's truly remarkable. One of the most amazing neuroscience books I've ever read is Wired by David Eagleman, and he takes that example to the extreme, where he's saying that neurons are so good at receiving outside information and converting it into some signal that's useful for the brain that eventually we could get a point where we're plugging Twitter or X into people and they're getting some sort of sensation or perception of, like, the geopolitical climate in, the other side of the world. Yeah, and if we can transduce any signal, and neurons are so good at basically converting that signal, we can effectively gain perceptions for anything. What do you think about that idea?
That's plausible but scary, right? So
yeah.
I mean, I'm partly worried that's where Elon Musk is going with Neuralink. I mean, his stated goal for Neuralink is having developed these wonderful flexible electrodes that can be easily implanted. So right now, of course, they're going to start trialing them in cases where that's really good. That's going to, for brain-machine interface implants into people with spinal cord damage and so on to restore some movement. But ultimately, the goal of Neuralink was to try and create an interface for any human with any machine, which relies on exactly that principle. Presumably he's read Wired too, which is exactly that principle that you can transduce practically anything into spikes. So we can interface exactly directly with we just have our newspapers and stream, instead of reading off the app, we just have it pumped straight into our heads and get that sensation of that headline is terrible. And we have people have played this a little bit, right? So we have people with try and added sort of sensors that we don't have onto people. So we can induce feelings of, say, people have tried using a form of echolocation and transmuting the echolocation aspect into some I remember this rightly, into some other. So I think the echolocation into a vibration. So then you can translate this into a vibration and have it then sensed by the person, even though it's not a natural sense that you've got. You can still interpret that sensation as information. And indeed, there's some anecdotal evidence from some people have been blind since birth that they have developed an echolocation like sense that they click and hear the echo and can roughly work out the dimensions of a room. So yes, it is kind of believably plausible that we can at some point it just stick an electrode into a brain, stimulate neurons to generate spikes that in proportion to some arbitrary information, that all it requires really is us finding a way of turning that information into spikes. And even then, it could be fairly arbitrary because obviously we can always be trained to respond, have our neurons respond in the correct way. So there's this fabulous set of experiments where they've rewarded a monkey in this case with juice if it can get one of its neurons that they're recording from in motor cortex to fire at a particular point in time, given a cue sometime before. So just some cue, make the neuron fire at some point. Without doing any movement, just literally make the neuron fire.
Okay.
And that neuron can be trained. The monkey can internally somehow get that neuron to respond at the right time. You can shift where it fires until it can reliably get juice. So we have this. There is having that kind of plasticity to get neurons to essentially to become informative about something which is entirely arbitrary, even though it's entirely self-willed, just by training.
Yeah.
So yes, although that then opens up horrible potential of
I want to get your thoughts. I want to get your thoughts on the neuroethics of it all later. I wanted to dive into Neuralink and get your thoughts on it. One of the most interesting examples that he details in this book is sensory substitution. So it's basically where he's built these high-tech vests. If you've seen Westworld, the show, apparently they actually use them on the show Westworld. But effectively what these vests do is it's converting data from a head camera, and then it's converting it into basically vibrational plates on some sort of vest. And then apparently after a certain amount of training, people are able to navigate around complicated buildings and structures completely in the dark because they have, some camera that is seeing the corners and everything that is sending that signal down to the vest, which is putting vibrational patterns onto the skin. The skin is transducing those signals to the brain. It's remarkable. Where do you see, where's the upper limit of that? How far could you play with that concept? And let's throw ethics to the side just for hypothetical and theoretical, play. How far could you push that with your knowledge of spikes, with your knowledge of the brain and plasticity? How far could you really take this sensory substitution, sensory augmentation? Where do you think all this could go in the next couple of decades?
That's a good question. So, yeah, I think we're already seeing obviously with Google Glasses and the Apple VR system, this idea of that we can have a system on our face which is pumping information directly into our eyes, but it's happening at multiple levels at once. So in some sense, it's a sensory substitution in the sense that you've been sort of act there. You've got all this information being streamed in, whether you want it or not. Anybody else to simultaneously respond to the world beyond the glasses and the thing that you're looking at right now, which is obviously requiring you to be trained to split your attention so that you can actually function like this. Apparently, people can. I'm not sure what is the upper limit? I mean, in my youth, I was as a big cyberpunk fan when that was all the rage of just the idea, sort of Bruce Sterling and William Gibson, all that, of the idea of augmented humans and how far that you could push this. And there, I would say, for all the creativity there, they were more interested in replacing bits rather than thinking about, what else could we add to the process? I don't know. So the limit, I guess we touched on already. The part of the limit is whether we want to add brand new senses to our repertoire, whether that be echolocation or a weakly electric field. You want to be like an electric fish and be able to detect small things coming within you. I'll see. Expanding our current senses would be straightforward. So given that we already have very advanced cochlear implant technology, it wouldn't be really that difficult to try and the microphone on those can happily pick up frequencies that we can't naturally hear. There wouldn't be any problem in theory with being able to hear pitches as high as dogs or cats or something as low as a blue whale if we wanted to. I guess it would then take some training to make some sort of sense of that. But then we can happily have glasses which shift our vision into infrared, and that's not a problem. So similarly, we can motion shifting is all kinds of other spectra that we can look at. But I guess, in terms of sensory substitution, the touch is going to be one of the most interesting ones to enhance, because we know a lot more about vision and sound than we do about touch.
Uh-huh.
But obviously, a huge amount of our sensory cortex is dedicated to processing touch on our fingers. So yeah, maybe it would be maybe interesting to see what's could be enhanced in terms of what we could feel through our skin. To the extent, I guess what a trivial example would be that you could imagine any kind of virtual surface being essentially projected internally onto the skin. So you wouldn't never need, say, sort of a keyboard, because you would just have the sensation of the keyboard played out onto the finger itself live. So you could just type.
Yeah.
Should we ever need to type? I mean, at this point, if that was possible, presumably by that point, the transcription from voice recognition would be by that point should be so good that you could just say what you wanted. Although coding by voice still is an absolute pain.
Yeah, I guess like the limits are I think underappreciated by people of what the brain could be transformed into, and I think that is a very, very scary thing, and a very, there's lots of ethical borders. There's cool examples, there's examples of painters having extra cones, so as you said, they can see more spectra of color. There's a few things where I'm like, that's cool. If we can expand art by just expanding the sensory system in some minor ways, that just gives us extra dimensions that maybe other mammals are already experiencing. I think that would be very interesting, but it's hard to know where you draw those lines. And the human race isn't very good at getting to a point and then saying, right, we've come far enough.
Sure.
Let's cut off the Neuralink advancing. Let's take this technology and not advance it. Like, what would you be worried about, I guess, in terms of neurotechnology developing in the next couple of decades? Where do you see real harm coming from?
So the big step is the transition between where it's compassionate use, where it's about use to restore lost function, particularly in, as we touched on already, particularly in severe. So cases of we want to restore hearing, we've got cochlear implants, we want to restore vision, we've got retinal prosthesis, we want to restore movement, we've got cortical and spinal brain-machine interfaces that can restore some movements, restore some walking. We've got recent wonderful breakthroughs in brain-machine interfaces for restoring some level of handwriting to paralyzed patients who can just essentially imagine writing a word in their handwriting and have it appear on screen.
Yeah.
And there's some new advances in language there too. But so that's all the sort of the easy stuff of going. So this is where people who have lost something want it back and are prepared to go through the, particularly for the invasive stuff like this motor cortex, spinal cord, restoration of movement, prepared to go through the invasive surgery and the many months required to train these things to get up running with them, working with them. In that case, the trade-off of the risk and the reward is clear. The barrier comes when we get to the point where we're going, well, we want to take this now, but we want to do it in healthy people.
Yeah.
So, I think Neuralink is a little bit a special case because they're very explicit about the fact that the end goal is to take their threaded electrodes thing they've developed, they want to put it into healthy humans. Yeah. Whereas almost everyone else that I know, I'm aware of neurotechnology companies developing interfaces for the brain, obviously primarily focused on medical use cases, and pretty much none of them is a truly invasive system. So Neuralink's is pretty much the only one that's genuinely invasive. There are others. There's the companies doing stentrodes, which is this nice idea of putting instead of putting the electrode directly into the brain, essentially just doing like a stent, so threading it through the vascular system up into the brain, so that you'll have this electrical electrode sitting inside the vascular, so you can pick up the changes around that electrode and essentially the local potential field. But you can't pick up individual neurons, of course. So that's both getting into the brain, but less invasive than putting an electrode straight into your.
And how is that inserted? How is the surge? How is that stent basically inserted into the vessel and then assumedly pushed into the brain or close to somewhere in the brain?
Yeah. So yes. So I'm not quite sure where the entry point is.
Yeah.
But clearly it'll be a similar entry point to wherever you do it. Sometimes do exploratory stents for like strokes, or where you got blood clots, you would go in through it into the veins. So possibly somewhere in the neck, wherever you got a good supply up to the head. I'm not sure.
Yeah.
But yeah. So it of course is so that's clearly less invasive, whereas the still somewhat invasive, but less so. So you run this whole gamut from a lot of people working on completely non-invasive stimulation techniques. So where you have the TMS or TDCS, where you've got a magnetic paddle hand over your head, and it's sending out a pulse of pulsing magnetic field, and that's doing something to the neurons inside the cortex, and that's creating some sort of response. So you can reliably get an arm movement over motor cortex. If you do that, you can do various things with it. It's quite a blunt tool. And then, you say at the very far end is Neuralink, with the idea that we want to go into healthy humans and stick electrodes directly into their brain so that you can record directly the spikes, and not only record the spikes, but somehow write back to them some information, which is then, as you're saying, it's the bit where we get into the sort of the science fiction realms of, well, what is it we're going to be writing back? What can we send these neurons? With as I said, the obvious barrier that we have very little knowledge of how any advanced concept in the human brain is coded in terms of spikes. So without that knowledge, you're just firing stuff in the dark. So as alluded to earlier, perhaps you could train the brain to respond, to recognize that it's being fed newspaper headline from a streamed app by rewarding the brain whenever it sort of recognizes roughly at the tenor of the headline. On the other hand, you can imagine a system there where you're being rewarded for a particular affiliation, but not the other one. And that very quickly can be
Explain hypothetically how that situation might happen. Like if we were trying to teach the brain to understand some type of artificial signal, like newspaper headlines or something like that, how would you reward the brain? Like even theoretically, like how actually would you do that training process?
That's a good question. So I guess as with all these things, you would scaffold them, right? So you've got you start. So everything would have to be reported by the person with that electrode in. So they'd have to report whether they could first start reporting whether they could detect the input or not. Just literally, is there something that's when we switch this on, can you detect it or not? So there'll be a whole bunch of obviously randomized blind trials there with where you do everything except send the signal. So all the switch goes on, everything, all the other things you could use as a predictor. So then once you've got past the first point where you can think, okay, there's a detectable thing, then you're trying to work out now you've detected it, what is the information it's trying to convey to you?
Yeah.
So it'd be interesting to see if so if, for example, you try to maybe what you did was you trained an artificial neural network, a spiking one in this case. You gave it the newspaper headline as input, and it spat out an output which was transcribing this thing into some kind of code, which, when you train a network, would reliably distinguish that headline from, say, a whole bunch of other different trained sets of words, at least in terms of, say, subject matter. Say this headline was about whales being stranded on a beach or something.
Yeah.
So then you could the next stage would then be to play in a set of these signals and say, can you tell the difference between this one and these ones? So does this feel like a different thing to you, this headline, as opposed to these ones? And again, randomize these, double blind, all that kind of thing. And I guess when you get to actual meaning, it would then be down to obviously that would be a bit more subjective about whether you're trying, but the sort of semantic response you want from the subject about the fact that maybe they want to at least tell you there's an animal in the headline, right? So that would mean, I guess, creating a whole bunch of headlines with got animals in them, having some kind of response screen, which was does the headline have an animal in it or not? So yes, no, in it. I mean, initially it'll just be, and you get rewarded every time you get it. Right.
And when you say reward, are you literally giving them a cookie? Or like what is the actual reward here?
Yeah, humans. That's another thing we can spend ages on. That's a side issue. Well, humans find so many things rewarding.
Yeah.
So obviously, animals, you have to give them something, right? You have to give them some juice, some chocolate milk, some chocolate hoops, some banana. Humans, you can flash up on screen that you've got some points, and they find that intrinsically lovely, right?
Gotcha. You just gamify the system with some reward feedback loop that doesn't actually have to be something ingested. It can be just, it makes sense.
Yeah. Yes. Humans just sort of love maximizing their return on whatever it is.
Yeah, literally.
You can always translate. Obviously, often this gets translated into actual rewards sometime at the end. Right? So the whole point is that you're trying to maximize points for a reason. Maybe you just like to be high up on the leaderboard, but often it's because you're told this will translate into some kind of monetary reward at the end. And somehow in humans that's enough. And this is a whole other area of mystery of how in animals we know very well how that sort of reward feedback system is working. So you give them an actual physical reward, and you know what that triggers. Eventually, often that ends up triggering a response in the dopamine system. But in humans, it's rather a mystery how sort of points appearing on a screen or a big flash sign saying you won or you lost gets translated into an actual intrinsic physiological response in the brain that gives an error signal.
Yeah,
but it's a long way removed from food. So it would be, imagine that it's something like, you would how much headlines where it's got animals in or not, you would do feedback about, yes, are you getting it? So, and that way you would then train in a sense of that sensation in my head means there's an animal in the headline.
Yeah.
And then you can then, having trained that, you should be able to be shown a whole bunch of your sort of classification performance should go up. Just thinking of humans as a big neural network, essentially. It should go up and up and up. And you can eventually see how that would then, you can nuance that. You can think, all right, a whole category of animals, then let's break that down to, all right, cetaceans and everything else. If you've got whales and dolphins as well. But it'd be, obviously training that would be wouldn't be a realistic way of getting deep neurons into a whole bunch of complex headlines, but would be the sort of an obvious first starting point for how you would get someone to turn an arbitrary pattern of spikes into a meaning for them, because you can start attaching their own internal labels to that.
Absolutely. So that makes a lot of sense. So it's about semantically labeling things in the real world, like animals in the headline, and then basically just rewarding someone yes, no on that true north signal, and then basically the neural networks are optimizing towards that true north signal of that's an animal, that's not an animal, and then you just keep breaking up those semantic labels and you keep training on these objective true norths in reality, and then for somehow that network is just getting more efficient at classifying that information. But you would need to do this for a very long time because neural networks, you need huge data sets just to recognize numbers, right? So I wonder what the human brain would take in terms of this. If I remember right, this is vaguely similar to when they were training people to use the vests. That's how it worked, in that they would show different items in front of the camera and they would say, is this a person? Is this a banana? And then it just kept going like that. And then those signals were again transduced to the vibrations. I think it's fascinating just simply that vibrations on our skin can be transduced into something like visual stimulus. I think that's phenomenal. But I think it's a very similar process. I thought that was interesting to dive into because it might give people an idea of how the neurons really are programmable and how that program happens through development with the different stimulus that we encounter. I guess an interesting next point to go to is the horribly tragic examples of when that stimulus is removed. There's some cases in history of, like, I'm sure you're familiar with the Romanian orphanages in I think the 1950s, 1960s, where they had a huge spike in birth rates, and then they were basically all put into orphanages because people couldn't take care of them. They were deprived of love, empathy, care, everything, and these, they just did not develop properly. Do we know what's happening in the brain when those neurons don't get that critical stimulus? Like, what's actually happening on that neuron when they don't get any environmental stimulus to be programmed by?
So it's a little bit speculative, obviously, but you're right. We know a little bit about about what happens to neurons in animals when I touched on it a bit earlier. Neurons in animals when we deprive them of various stimuli. So, for example, we know a little bit about how from early experiments in the 60s, we have an animal raised with one eye permanently shut, so that the signal coming from two sides, obviously, crosses over in the brain, and that's pre-wired. And the question was, right, if that's shut, does that side of the brain know anything about that side of the world or not? And the answer was obviously that it didn't, but the wiring was still there, so it could. So if you then unclose the eye, it could rebuild a map of the world. So we know, yes, there is definitely through development and experience of the world, that missing various experiences of the world means that various neurons we expect to appear with particular functions don't get those functions, even though they have potentially the wiring to do so. So that's really clear for simple sensory systems. Also, when we get to more the more complex cases of more what happens in more social environments. So as you say, that sort of Romanian orphanages were a classic case of extreme deprivation during childhood, and all the problems that caused. But it's done quite a bit of guesswork as to exactly, we can take the same principle and sort of guess at the fact that given that there was an absence of any sort of physical contact, that there was the parts of the brain that are more heavily involved in social interactions also be missing the stimuli that they were expecting to get. So we've seen this a bit now with current modern antenatal care is for newborns with their mothers is very much emphasis on the skin-on-skin contact, very straight after birth, right? Which is sort of thought to have all kinds of benefits. One is just not least the getting the newborn baby to generate a better microbiome in its gut by getting some of the bacteria off the mother's skin. But it's a lot to do as well with the bonding process of that skin contact releasing various hormones which help generate a sense of bonding. And obviously, ultimately, that sense of bonding is carried by neurons firing that are saying, here's the sensory signal coming in, and this is a good thing. So extrapolating from that, then obviously in the case of those extreme deprivation, clearly those Romanian orphanages, they're missing all of this contact, any of this thing with this physical contact with other humans, all the richness of the environment that you're required to develop properly, and then you've clearly even though presumably the neurons exist, they're not wired up to respond to the thing they should be responding to.
It's interesting they're still there. So if the neuronal wiring is still there, but it seems less responsive or completely unresponsive to the stimulus that you would expect it to respond to, are there cases where you can retrain those neurons? And before they're retrained, what are they doing? Are they just kind of sitting there idle? Are these the famous dark neurons that you talk about in the book a little bit, which I definitely want to get to as well, because I think they're really interesting. So what are those dormant neuron wiring in the brain? What are they doing?
So let me see. So yeah, no idea whether
questions in there.
Yeah, don't know whether or not they're the dark neurons. That's yes, they are. They're possibly a separate group of neurons. What often happens with neurons that are not used, so they exist and they're not used for a function which, in theory, they're what in a fully intact brain they would be used for. Often, of course, they are then co-opted to do something else. Right. So, we have classic knowledge of people who are congenitally blind. So their visual cortex appears to shrink quite a bit, and that cortical real estate is taken over by other representative senses. So whether it be touch or sound or whatever. So in other cases, yes, it may be that some aspects of the brain have atrophied. So while the actual sort of neuron numbers as far as we know, tend to be genetically specified. Obviously, the richness, the connectivity that they have tends to be driven by experience and development. So, so there's great early experiments on raising rats in impoverished environments. You can raise a rat in a really rich environment full of toys and things to play with, or you can just raise it in a plain cardboard box. And you compare those to two groups of rats, and you can see the ones in the rich environment have much thicker cortexes. They have much more wiring. You can see more white matter, so more myelin around the axons, which means that signals can be sent faster. The brains just look as though they have are much more richly wired, or much more able to respond to their environment. So, whether they can be recovered or not, in many cases, in principle, yes. Right? So we know, there's lots of work on neuroplasticity already in humans about how people can recovery after stroke of language or movement, because the area has been damaged, that function has been co-opted into a different area of the brain. So if you can do that, then presumably you can also take intact tissue and repurpose that purpose it back to what it was originally useful supposed to be.
Yeah, it's incredibly interesting. So back to this atypical development of what's happening when the stimulus isn't being received. Can we see anything physically in the brain? Is there any type of scan? Is there any type of, if you biopsied that tissue, what does it physically look like? So you mentioned less branching of those neurons onto other neurons. Are there any other physical characteristics that are dependent on that developmental stimulus?
So to a large extent, it's so what we can see in sort of obviously animal experiments. Yes, we can see in detail something about the way that the wiring between the neurons depends on the stimuli. So, for example, we know in the healthy rodent brain that if we look in the early parts of visual cortex, if we see a neuron that responds to an angle of, say, 45 degrees, then it's more than all of the neurons around it that's going to be connected to. It's much more likely to connect to another neuron that has a preference of 45 degrees. So that is all worked out in development too. That this sort of like for like connection between these neurons.
Mm-hmm.
So a lot experience imposes a lot of structure onto the wiring that we can see in detail in animals. That one would guess that in the absence of those stimuli, that wiring is essentially is random. In fact, there's a good one model of how the brain, particularly the cortex, develops is that all the wiring that the wires that the neurons send out start essentially connecting to things at random. They create physical contacts, so there's touching. But whether they get turned from physical contacts into active ones, as in ones which are actually a synapse which sends transmitter across the other side, that's largely driven by experience about what it needs to connect to in order to make best sense of the world. So in animal models, we can see this in great detail. In humans, it's a lot harder, of course, but we can do, as I was sort of alluding to, we can do, for example, MRI scans to look at the anatomical structure. We can see how much white matter there is. We can see how thick the cortex is, for example. We can see the differences between different brains. And presumably then you would see in humans, for whatever reason, have a highly deprived development, you would see less white matter, less thicker cortices, and so on.
What does that tell us about how the environments that we should be putting children in when they're developing? Obviously stimulus rich, but is there any more nuance that we can add to that in terms of the stimulus that children should be getting, and beyond children as well, and even in older people? What sort of stimulus can they have to prevent that cognitive decline as well on the other side of development? How does this knowledge teach us about what environments we should be putting people into for optimal brain health?
It's a good question. So, I mean, I think those quite a lot of those early rodent and primate experiments, so from the 60s through to the sort of late 80s, on the type of social environment you raise animals in and the type of richness environment you raise animals in, has fed into now our current way that we approach raising and looking after children. You go into any sort of modern kindergarten or nursery school, and it's just stuffed full of sensory stimuli. It has sensory rooms, it has clear, quiet areas, it has touch areas, textures, sand, play, everything, all kind of textures and to grind create this as a rich environment as possible so that you have this, so that you both have a chance not just develop the basic senses, but also to explore your capabilities as well. So if you turn those that as a large part of development is getting your motor skills up to scratch. So having a chance to explore these different environments and be able to touch all this stuff and then what you can manipulate and what you can't manipulate is a large part of the richness of the world. So, I would say that sort of extent has already fed forward into culture about the way we approach childcare. We hear it a lot. We've known now for a couple of decades. Things about language acquisition. Right, there's sort of the window for when the brain is most plastic, when the neurons are wiring up and they're most plastic to be able to cope with the different phonemes that go with different languages. That window is lasts for a couple of years, maybe a few years, and then shuts down again. So that if you want to have a genuinely multilingual child who can actually pronounce some of the phonemes in languages, say Japanese and Gaelic, at the same time, then you have to expose them as early as possible. Otherwise, it's going to be later in life. As anyone who's tried to pick up a language later in life knows that pronouncing some of the stuff properly becomes a matter of sort of acting, imitating it, rather than being able to do it naturally.
This is something I'm reminded of every day living in Spain and trying to speak my awful Spanish, to which they always immediately respond with English. So this is something I'm reminded of on a daily basis.
Yeah, yeah, indeed. But so at the other end, you're right. So the other end of the spectrum is what happens during aging. And I always say we know a lot less about what happens to individual neurons and spikes in the brain as we age. We know obviously a lot about like cases that we know a lot about dementia. We know a lot about the obvious cases where the brain is failing because we can see on a brain scan and we can see big holes in it. So the neurons are dying en masse, or you're losing all these connections, and clearly the cognitive function is declining. So we know that what that means that underneath that is that there's a whole bunch of neurons, A, we've lost that just aren't there to fire anymore, and B, that haven't got the right connections to where they need to be, so their function is no longer happening. So we know that ultimately the sort of the loss of these spikes being passed around the brain, it's underpinning this cognitive decline, but how you map that back out to an intervention is very hard to think about. Hence why most of the ways we think about sort of about preemptively addressing our worries about, say, dementia or other cognitive decline is all about just making sure we stay stimulated, making sure that our neurons are active, told to keep cognitively engaged. So if you are retiring, go and do some crosswords, go and help out a local community centre, go and volunteer and do something, stay active, do things. So the whole idea of, treating the one aspect that's always talked about is treating the brain like any other muscle, just keeping using it so that it will stay healthy.
Yeah.
And yes, I mean, and so exactly how that would work in the brain is unclear, but presumably it's related to the ability of the brain to clear out damaged, misfolded proteins and all the other garbage that collects. And you need to get rid of it. And clearly, we know that from development through adulthood that just this whole idea of the more a neuron is active, the more likely it is to maintain connections to other neurons. That's true. So it makes sense just from that perspective that we carry on engaging in a rich variety of activities and things that are based on cognition. So we make sure that we try and engage as much of our brain as possible, and that's going to hopefully have some kind of protective effect against the accumulated damage of aging.
Yeah. I've thought a lot about how you would, from the ground up, like optimally design a old folks home if a bunch of neuroscientists were in charge. And with infinite money, how would you make it so that these brains decline the absolute least across time? You'd have like table tennis tables, you'd have loads of different things to not just exercise your physical muscles, but also exercise every single aspect of the brain. Like I guess ideally you would want to be flexing as many different modules as possible so that they're getting good exercise, so that they're reading, so that they're listening to music, so that they're sewing, they're touching textures, they're seeing colors, they're trying to basically barrage the sensory system and physical system with as much information for as long as possible. And as cruel as it sounds, kind of give them the least amount of help so that they do as much by themselves, so they don't lose that agency, they don't lose that autonomy. These are really interesting things to me, and I don't know if there's been enough input from neuroscientists and what we know about cognitive decline in how to really rebuild these places where old people go to, and they seem to just have a very precipitous decline in their cognition. Have you ever thought about that, or what do you think about those different ideas in terms of like how to keep them as young as possible?
That's a good question. Yeah. So no, I haven't given that any deep thought yet. Perhaps I'm just pushing it away.
I don't know why I've thought about it, but this is like a random Saturday thought for me.
But I mean, you're right. There's a sense that what you're describing obviously is a little bit like essentially taking all the stuff that we have developed for all our sort of kindergarten, nurseries, the deep rich environments there, and taking a whole bunch of that and basically sticking it at the other end as well, of making sure that there is just a wide range of things that to be engaged in, that engage all the senses, that are not just end up sat down on a chair watching TV or streaming or whatever. Yeah. Yes. So I guess it's going to be as we know from any aspect of making sure humans develop and stay healthy, it's going to involve not just a rich environment, also the social aspect is key, right? So making sure people are interacting well and constantly. And yes, it's unclear to me whether presumably there are. I mean, I remember seeing an advert just the other day. There's a new, very advanced-looking care facilities opened up just about a mile from us, all full of glass wall apartments and all this very beautiful-looking thing. And I remember wondering how rich the environment was inside. It looks really shiny on the outside. Is it actually as engaging on the inside? Is it full of plenty of different rooms where people can go and socialize? Is it full of Yeah, it's a good question. Perhaps we should give some more thought to, yes.
Yeah.
How rich the environment needs to be at the end of life as well as the beginning. Yeah.
I think it's an interesting problem and obviously an important one. If the power of the brain hasn't been clear up until this part of the podcast, which I think it really eloquently has been, there's a great quote that I liked in the book, The Spike. Each neuron in the cortex is approximately a computer. So to think that we have 86 billion approximate computers in our brain is quite phenomenal. How do these neurons compute logic? Like, how does logic work between neurons? Because I think this is another fundamental thing that people probably don't understand fully, but could really help them understand how this processing unit and this lump of fat in the brain really does what it does.
Right. So there's let's pull those things apart. So the idea that an individual neuron, particularly here, this pyramidal cell we mentioned earlier, this big cell in the cortex with this triangular body, this big axe, the dendrite sticks out at the top. There's a lot of really nice work, some from Piotr Perazzi back in the early 2000s, about showing that you can take one of these cells and you build a detailed model of it. So you simulate its body, but also a big chunk of its dendrites and the way the dendrites branch and how the inputs arrive on it. Then what it seems to be, that an individual neuron like that can act as what artificially would be a multi-layer neural network. So it does that because the branch of the dendrites essentially are independent of each other. So they can individually sum up their inputs, which gives you one layer. And then they send that down to the body, which is your second layer. So minimally, you have this two-layer sort of neural network type thing. And the two-layer neural network can compute all kinds of interesting functions. So as you mentioned, you've got this idea of neurons computing logic. So one place that's come from is because one way we sort of quantify how well something can compute is we can ask it, how many logical functions can you do? If I give you a set of inputs and I want you to give me a 0 or 1 as an output, as a true or false as an output, how many of these things can you compute? So the more you can compute, the bigger your computing power is going to be, the more complex things you can do. So I mean, that's how we think about how computers themselves their processing depth is really, how complex a logical thing can you compute? Because after all, pretty much every computational statement is about, it ends up being some kind of true or false statement. It's about from just adding numbers together to doing some complex thing. Is this a biscuit or not? Classify. Ultimately, this complex function ends up with a binary output. That binary output is used by the next operation along to do the next thing. So we have this idea then that the neurons are these individual neurons can act as a multiple layer network, so they can compute fairly advanced functions themselves. And then what they do with that is a good and open question. So, but it is true that a lot of the history of how we think about what neurons do is driven by this idea that they compute some kind of logical function. So we had the most famous paper, sort of probably the first proper sort of theoretical neuroscience paper from Warren McCulloch and Walter Pitts back in 1945. But the idea is it was showing how if you assume that neurons either have spikes as outputs, have zeros or ones as outputs, which already speaks to binary, so it speaks to some idea of logic, and that you can then you can then show that you can find any configuration of neurons with just addition and summation and subtraction put together. You can compute pretty much any statement in logic that you want. So that means that from that point of view, that you could think of the brain as constructing a series of logic statements, and therefore computing, much like a computer, in a sequence of if this, then this, if this, then this, and so on. Of course, that's not, as it turned out, true. There are lots of operations in the brain which don't follow a statement of simple logic, are more analog in the way that they work, not least the triple example of how we started with muscle contractions. So obviously, a muscle contracting is not a logical statement. It contracts in proportion to the number of spikes that arrive at that muscle. So the more spikes arrive, the more contracted it will be. There's no logic going on. It's an analog signal saying, I need you to move this arm this quickly, so I'm going to send this many spikes that quickly. So much of the brain appears to be this more sort of analog, so it'd be like coding scheme. Nonetheless, the sheer idea that collection of neurons could compute in a logical form gave rise to the whole lots of ways of thinking about the brain that were new about how it can compute. And there's still quite a lot of interesting research on how in an individual neuron, a particular alignment of inputs can help it compute things like an and statement between this input and this one, and I respond not to them individually. So you've got a clear and on there, or you can have an or type statement where one or two inputs is responded to, but not both together.
Yeah, it's fascinating. It's so interesting. So that sounds a lot like a computer. What ways do the brain and the computer fundamentally differ in how they compute logic and how they compute signals, how they compute functions, anything? What are the biggest ways for an average listener where it might have a misconception about how the brain is related to the computer or computer is related to the brain? How do they fundamentally differ?
So this is a deep hole for most theoretical neuroscientists. So it depends on the definition of computer. So the computer that's sat in front of me now is a keyboard, screen. It has a hard drive. It has a processor. It has a memory in it, so on. So when you say computer, it most often invokes that kind of architecture, something which has got a processor in it, has got a memory in it, it's got some kind of interface system. So whether it be my phone, this laptop, a tablet, a really smart fridge, that's the way that we think of computers when we say computer. From that perspective, it's very easy. The brain is that case not that kind of computer. Right? We don't have a separate memory store from a separate computational store. And then there's the more complex version, which is a computer is, if you say a computer is sort of Alan Turing or the George Church type approach, where we say a computer is anything that can run an algorithm, then the brain is a computer because it can run pretty much any algorithm. But in that case, a rock is an algorithm because it runs some physics algorithms. It runs falling down when it's released. Right? So in that sense, the brain is trivially a computer, but not interestingly a computer. So somewhere in between is the interesting question of if I commit myself to a particular model of how a computer works. So, for example, if I say I really want to commit to the idea that the brain works by say, yeah, okay, let's start with you mentioned logic. So brain works by having a series of statements of logic, which is the same as we meant inside our laptops or our phones in the processor, where you have a series of transistors which are either on or off, so everything is in this binary logic form. Then the sense in which it's the same is the sense in which, yes, neurons have this binary output, this 0 and 1, this spike. That much lines up. And indeed, historically, that's what inspired von Neumann when he designed the original sort of what's now the architecture that we use for everything with separate processor and memory and the way it's controlled, the buses and so on. He was inspired by Culloch and Pitt's work on the neurons and logic and the fact that we want something that has a 0 and 1 because that will work really cool. Hence, we have a system that's based on this 0-1 idea. But as we just noted, it's very easy to find ways in which the brain doesn't work in this simple binary logic way, in which you have a lot of particularly the work of the neurons towards the periphery, where they're controlling the muscles, or the input they get from, say, the senses, isn't done by logic, but it's done by transducing this information into some kind of analog signal that is used by neurons further in.
So what about artificial neural nets versus brain neural networks then? What are the main things that those systems do that are the same? And then what are some of the fundamental differences? Because, neural networks are all the rage at the moment. People talk about the large language model development in the last year or two has been an absolute explosion. I think people have this word neural network in their head and they sort of understand that's how neurons work in the brain. And that's also how ChatGPT works in large language models. What are some of the parallels? What are the similarities? And then what are the big crucial differences, if there are any?
I guess the main similarity is that, at heart, in an artificial neural network, you've got a whole series of units, and those units they receive input from a bunch of other artificial neural units. They sum them up, and they give some kind of output as a response. That's the fundamental operation in any artificial neural network, and that is a true, if crude, cartoon of how actual neurons work. So yes, neurons get their inputs, they summate them, and they turn them into some kind of output signal. So this kind of spikes. I mean, the principal difference is that pretty much any artificial neural network you hear about or read about, of course, the output is just a continuous signal. So all these inputs, these units are getting, their outputs they're giving, they're all continuous in some way. Often they're bound between 0 and 1, but in that range they are continuous. Whereas, obviously, what a neuron sends out is a spike. So the only way that the way they translate as being the same is that only if we have a scheme where we count the spikes and say, all right, so if a neuron can send out something between 0 and say 100 spikes, and it's currently sending out 5 in this second, or maybe it's sending out 10, maybe it's sending out 20, then you have this continuous range of the number of spikes. But there's good reason to believe that a whole lot of processing in the brain doesn't work by counting of spikes, because often there just simply isn't enough time to do that. So, so there's some nice famous experiments where they look for, they're flashing an image up on the screen and looking for the earliest detectable response to the image on screen inside the prefrontal cortex. And the earliest detectable response happens within about 100 or so milliseconds. So 100 or so milliseconds, most neurons in the long chain from the eyeball through the various connections in the brain from the visual cortex all the way back all the way to the front, most of them haven't had a chance to send a single spike, let alone multiple spikes.
Yeah.
So however that information is going to the front, it's not because every neuron is waiting in turn for saying, oh, I've just received 20 spikes, I'm now going to send 10, go down, because that would take forever. So there's a clear reason why so a fundamental question really actually in neuroscience is, why does the brain use spikes and not something else?
Yeah.
Given that we can happily design artificial systems which don't need them. So artificial neural networks can do really cool stuff, but don't need them. So it's an open question as to whether the reason the brain uses spikes is because of some fundamental biological reason. So, for example, that to send information biologically over long distances, you need a system where you can just send a pulse that is reliable, that you can regenerate, that will travel a long way, because sending a continuous signal will dissipate, it will fall apart, you'll get lost in noise very quickly. Or whether there's a fundamental computational advantage to having the spikes, in which case, whatever that is, that's missing from artificial neural networks.
So that difference really isn't quite known just yet of why it's using a discrete signal versus a continuous signal, and what the benefits might be of that specific spike. Because the brain does seem to be, and you touch on this in the book as well, it's far more energy efficient than any artificial neural network, right? Magnitudes more energy efficient. Could that be because it's processing a discrete signal versus something that's maybe bulkier, which is a continuous one?
Yes, and certainly that. So yes, although of numerous reasons why it could be using spikes, the energy efficiency argument is a really strong one, and that's indeed the argument underpinning why so many of the big chip companies are trying to develop neuromorphic chips, where the processing on board is essentially spiking neurons as opposed to transistors, because you can then in those chips, in theory, do a lot of similar computational work, but vastly lower expense than if you're doing this continuously updating thing, essentially because it's an event-driven system that you only ever bother updating when a spike turns up.
Explain that a little bit more. How are these neurons existing? What is a neuromorphic chip? How are these neurons existing on some sort of motherboard or something? Explain that a little bit more.
So in the ones that I know best, they essentially build into hardware a particular model of a neuron. So there's a particular class of model neurons I work on quite a lot, that some neuromorphic chips embed directly into them, essentially create layer upon layer of these types of neurons. And then I guess we're back to the logic. They end up being able to obviously use them to be able to do traditional computational operations on them, but now simply by passing spikes rather than having millions or billions of transistors doing the same job. So there's still a lot of money being pumped into these things. They've been going for neuromorphic chips, a good 15 years, probably 20 years now, and are still big announcements every now and then from, say, IBM or Qualcomm, and saying, look, we got this amazing new chip that can do all this new stuff. But yeah, it's still not clear to me how much further they need to go forward in order to be usable, whether we actually cracked the sort of the energy efficiency barrier.
Gotcha. Explain. Let me know if I have any of this wrong, but it's my understanding as well that one of the crucial differences between the artificial neural networks and the neural networks in our brain is that backpropagation is a very important process in which the output of an artificial neural network is fed back in the layers and adjusts the layers. But there isn't any backpropagation process in the brain. You know, the electronic flow goes through the axon in one direction and doesn't flow backwards. Is that accurate?
Yes and no. So you're right. So obviously the way that backprop works in artificial neural network, it's done so in a way that it's very hard to conceive of how you would do that biologically. Essentially, because in the way that that's done, obviously that's, as you say, that's taking sort of the output of one layer, sending it back up to the layers behind it to essentially tell them where the error was in what its output was. And to do that, it needs to know something about the change of the weights that happen behind it. So it has to transmit backwards information that you couldn't possibly know in biologically speaking. Having said that, there are mechanisms by which neurons communicate to the neurons behind them.
Uh-huh.
So when a neuron, some particular types of neuron, not all neurons, but when some types of neurons, like the pyramidal cell, when they send a spike along their axon, because that is initiated right at the edge of the neuron's body, that also there is a similar depolarization of the membrane that gets propagated back up, so it gets sent back up into the dendrites. So there is a sweeping back electrical signal, and there's a lot of speculation, a lot of work on how that might drive maybe a signal for driving changes of the strength of connections between neurons. So
Gotcha.
that when that signal goes back up, essentially the signal to the rest of the neurons saying, I have just sent a message. If I have received something interesting just before that, maybe it's worth changing one of those synapses. And there is a whole little sort of cottage industry of theorists who have been trying to work out ways of making something like a backprop work plausibly in neural tissue. So, yes, so there's a bunch of schemes for making something that looks like something has to know about the change in weights in the layers behind it work. Some which are more plausible than others, but yes, it's an area where people feel like, okay, so it's not entirely impossible that a brain could do something like this, but clearly not in the exact mathematical way that backprop works.
Gotcha. That's great. That's updated my understanding, because I wasn't sure that there was any resultant feedback structure that sort of resembled in some way that propagation. So that's very interesting. Let's jump to dark neurons, because these are really interesting, and this is probably one of the biggest things that shocked me about the book when I was reading it, because I just didn't know it. I did a neuroscience degree. I read about neuroscience like almost all the time, think about neuroscience almost all the time, and this was just a single statistic that I'll let you say that just kind of shattered my reality of how have I not read this before. So let's give a brief description of sort of dark neurons and kind of what they are and your work around those.
Sure. So simply put, it's the idea that the majority of neurons in the brain, particularly in cortex, are not sending signals most of the time. Yeah. So our view of the brain is often that we think about a neuron, it's sending its active potential, sending its spikes out, and that's how it's communicating with the rest of the brain. And when we, as you say, do like a neuroscience degree, read lots of neuroscience papers, you lower electrodes into the brain, and they report all these spikes coming from all these neurons that they record from, and that is the basis on which then theories and ideas about how the brain works are based, because that's where the spikes are coming from. But the problem is with those approaches is that, of course, if you record spikes from neurons using electrodes, then, of course, those electrodes will only pick up the neurons that are spiking. By definition, it can't pick up the neurons that are not spiking. So a long time ago, particularly in the 70s, and back-of-the-envelope calculations said, shouldn't we be seeing a lot more spikes? Because we know how densely the neurons are packed. We know where the electrode is. That an electrode in the middle of a rodent cortex should be seeing something like 100 neurons, and yet we're seeing the spikes from 2, 3. And indeed, it was kind of a matter of relief, really, that because if that wasn't happening, then the recording from the electrode would be useless, because you couldn't tell which spikes belong to which neurons, because there would be so many spikes that it would just be a muff of signal. So roll forward a couple of decades, and we've got lots of ways of recording neurons where we can see the neurons and record them at the same time. So we know exactly which ones we're looking at, which are active or not. Various imaging ways. We can also now have various very delicate ways of attaching electrodes directly to individual neurons. So we can we know the neuron we're recording from and ask, are you sending any spikes? If we do both those things, then we see that most neurons are not sending spikes most of the time. So it's a roughly a sort of classic sort of 80-20 rule, that 20% of the neurons are sending about 80% of the spikes, at least in sort of surveys of cortex that we did a fair few years back now. And so this then that other 80% of the neurons is what I sort of labeled as the dark neurons. These neurons that are traditionally sending much less often than one spike every second. With imaging stuff, we can stare at neurons on a camera, see a bunch of them flashing occasionally, and there's others that are just sitting there doing nothing. So, and as I know in the book, this is a real pain, right? So it's a real pain because most of our theories are based on our recording some neurons that send spikes. So we have based on our theory. So what these other neurons are doing and what they're for is something of a deep mystery. So there's been long been sort of little lines of work on this. There was a famous paper little discussion paper by Bruno Olshausen called "What's the other 85% of a V1 doing?" So the first visual area of cortex, he had a little book chapter was going, well, back of envelope calculation says, when I put electrode into visual cortex, I'm recording only a tiny fraction of the neurons that are sending spikes. And then of those neurons that are sending spikes, most of the ones recording with these electrodes aren't responding to the visual world. Right? So your classic sort of work from the 70s and 60s that Human and Wiesel got Wiesel got the Nobel Prize for, is where you put your electrode in, you've got this neuron that's responding to an edge 45 degrees. That's what it's tuned to. But of course, there are other neurons around it that no matter what edge you show them, they don't respond. So they're sending spikes, but they're not responding to this edge. And there's loads of others which aren't sending spikes. So his point in that lovely little book chapter was going, right, so my calculations are, we know about what 15% of the neurons in visual cortex are doing. There's the rest of it, which is the bulk of it. We have no idea what it's doing.
Yeah.
And now we have a much better idea. So now I think that's this understanding of this darkness happens to have coincided with a shift in the way that we think about how the brain encodes information and how it computes. So now when we have these amazing studies where we use imaging to look at thousands of neurons at the same time, if not tens of thousands, and say we do that in, say, visual cortex, and we flash images at the animal, and we can see this huge area of brain. Some of the neurons light up, some don't. But if we take all that neural activity together, that single snapshot of response, then we can very easily sort of tell what image was shown to the animal. So across the entire population, the information is really, really clear.
Uh-huh.
So this, partly driven by this dark neural question, but partly just driven by technology, we've got this shift into this idea that we need to move away from understanding what individual neurons are encoding, because so few of them are actually encoding stuff that we need to know about. What we need to know about is what all of them are encoding. And in that case, even if they're only sending one spike every now and again, if they're sending that spike along with the rest, sending it with a whole bunch of other neurons sending them at the same time, then there's still a lot of information being transmitted out. It's just not being done by each individual neuron sending a lot of stuff.
Yeah, it's such a strange conundrum. Is there a chance that those dark neurons have a role, they have a specified job, we just don't know what it is, and when we're recording from those electrons, we're not presenting it the right stimulus that it knows and it responds to? Like, it's a ultra-specific stimulus that might, maybe we don't even understand how it works in the visual world, and it is responding to something, and it acts in a very specific manner, and it's just not relating to any of the visual quantities that we have discerned, like edges, corners, light, these different things. It could be just something else that we don't even have a word for or something,
Yes. So that's indeed. So one of the main ideas about what's happening is that in any given lab environment, lab task, we're giving an animal, we simply don't give it a rich enough set of stimuli to engage most of these neurons care about. Yes. That's one option. So when this will become clearer, I guess, as we get more and more into people who are able to record for neurons for a very long time in various animals. So there's some great work being done on long-term recordings in zebrafish, for example, where they can record for many days and over a period of weeks. So one option indeed is that, yes, they're not responding to not giving enough stuff to do. They're not responding to the world, particularly in early visual areas. Yes, I mean there are other theories, but it's certainly an active area of research right now. So there was a paper a couple of months back in Neuron, which was almost explicitly about trying to find out what the some of these dark neurons were in visual cortex. What they showed in that paper was actually they are some of the neurons they found which weren't responding were being forced to not respond. What they were showing was that when a particular stimulus comes in, the inhibitory interneurons in the V1 were responding to that stimulus and suppressing a very large number of neurons.
Gotcha.
Where they were pretty much silent. So there's huge silence waves. And when they remove that inhibition, they can see all these neurons suddenly pop up in response to the stimulus that was coming in. So they weren't particularly tuned, if you like, to that stimulus. They were essentially noise. So they were being deliberately held down. So they're their suggestion was, at least in this little bit of visual cortex, this little bit of processing, the reason we're seeing so many dark neurons there is because they are deliberately trying to increase the signal-to-noise ratio, essentially.
Yes.
But then, yeah, there are many other theories about why this would work.
Lots of mysteries. Curious. Would there be any correlation between the distribution of my dark neurons and yours? Like if you were to look at the brain and you could somehow, have some map of what neurons are the active ones that are firing almost all the signals, and then all the dark ones that aren't firing any. Would there be any correlation between people, between males, between females? Like is that pattern, does it have any consistency to it?
So on the one hand, we say we would have no idea at all. So given that, all we can really say is about is that there are different types of neurons. So it's pretty clear. So obviously, I already mentioned the sort of these interneurons that sit in cortex. So some of the types of interneurons are much more responsive to their inputs than are the sort of dominant cells in cortex, dominant pyramidal cells in cortex. So we expect a lot of these higher firing neurons are these interneurons that are on this long tail. So they're generating a lot of the spikes. So there's so in that sense, then yes, similar brains will have sort of similar distributions of the spikes amongst the types of neurons that we expect, that the silence will be dominated by the pyramidal neurons, and the active ones, more active ones, the interneurons. But say within the pyramidal cells, there need not be any necessary relationship between, there is not least because it'd be very difficult to even think about what sense are we comparing them. So if we're just looking at the whole of V1, say, then yes, there's going to be this sort of rough same mixture. If you drill down to the level of say, column of cells which is looking to the top left of your visual field, would we have the same distribution of sort of excitability and silence? Well, quite possibly. But there's no sense in which there is a one-to-one mapping of cells, because we'll have quite a lot different number of cells in each little part. The reason they'll be wired up quite differently.
Yeah, of course. Gotcha. That makes sense. This has been such a pleasure, and I think such a great masterclass in neurons and how they really communicate all of the magnificent amount of modalities and in how we experience the world and everything. And I think you've done a great job at elucidating what we know, what we don't know, how much stuff we don't know, and sort of all the future research directions as well. I'll leave you with one final question that I like to ask every guest. So we're talking about the next decade in neuroscience, and I want to know, what are you most excited about? And then the inverse of that is, what are you most scared about? So anything in neuroscience, neurotechnology, AI overlap with neuroscience. So just very broad category. What are you most excited about? What are you most scared of?
What I'm excited about? Most things that excite me are really quite nerdy, quite techie, actually.
I wouldn't expect anything less.
No, I mean from the perspective of someone like me who's so deeply interested in spikes and how they work in the brain and what they do. Is that the key thing that I really want, technology I want working for experimental science at least, is I want to see a thing called voltage imaging really working, which is
voltage imaging. Interesting.
Which is where so all the imaging I've mentioned so far is all about looking at the calcium inside cells, and that's a great proxy for activity, but it's not directly the activity. We've had the ability to have neurons express a fluorescent protein that responds to their voltage for quite a while, but it's only just recently in the last few years got to the point where that's worked well enough that you can now use it in a mammalian brain, in a rodent brain, for example. And even then, it's still only a handful of neurons at a time, about 30 neurons at a time, and it lasts for about an hour before it sort of stops working. But the advantage here is that you get the best of everything you want to be able to understand how the brain works. You get to look at exactly which neurons you're recording from, not least because you can create a genetic construct which says, I only want you to express this sensor, this fluorescent protein. I want to express it in a certain type of cells. So you can inject that and express it in a certain type of cells. You're looking at, say, the pyramidal cell in the hippocampus. You're looking at the interneurons in amygdala, wherever. And then because they're voltage sensitive, it means you're recording directly the spikes that are coming out of them. So you can see them You know exactly, and you can see the neurons. And the pipe dream is that somewhere 10, 20, 30 years down the line, that the signal to noise ratio will be good enough that you could actually see enough of the voltage itself, the bit that happens before the spike, that you could see some of the effects of the inputs to the neuron happening. So you could see the little deflections of the voltage that happen when the inputs come in. And then with that, you'll have this complete picture of the activity in a bigger network as you want to record from. So you can then go, great. So we know not just the spikes coming out, we know which neurons they're coming from, where they're coming from. And in principle, go a long way to inferring the detailed wiring between all the neurons. So you can start rebuilding the exact circuit that you've looked at. And then you understand its computations in a really deep level, because you don't understand not just what its output is, but also what it's sending amongst itself.
Gotcha. That's a really cool answer. I love hearing about the type of technology that researchers would like to see developed because of what questions it would allow them to start tackling. So really high resolution voltage equipment just lets you to tease apart these different brain cells and the exact cause and effect that's happening in those systems.
Yeah.
Very cool.
It's cool.
And what are you most scared of?
Yeah, what am I scared of? That's a good question. I mean, obviously, so keeping this on a neuroscience basis. So I think I'm not scared as well. I mean, I'm a little, I guess, worried that the neuroscience is particularly kind of neuroscience that where I'm interested in, the level of spikes, is that it's still in this. We're still a little scared that we're staying too long in our stamp collecting phase, and that it's still just the technology is getting better and better and better and better and better. And most of what we're doing with it is we're just collecting more and more and more data. It's just going, okay, we're now going to record from 100,000 neurons. Here's 100,000 neurons. Great. That's just 1,000 times more difficult to analyze than it was 10 years ago.
Gotcha.
But without addressing a particular question. So there are hints of good theories turning up, but I'm kind of worried to spend too long there, and that at the moment there is a big movement towards comparing, as you alluded to earlier, comparing the sort of activity from lots and lots of brain neurons to artificial neural networks, which is a really fun game to play. So, say, okay, so if I take an artificial neural network that's supposed to be processing images, and I go down it, say 1, 2, 3, 4, 5, 6, 7 layers, and I go into what I think would be the equivalent part of the brain, so maybe 3 layers in because of the extra processing, and I compare what the neurons are responding to, and they look kind of similar. Well, that's quite cool, right? So I've got this idea of what kind of processing happened in the brain. So that's a fun sort of analogy game, but I'm worried about how far you can. People are getting very excited about this at the moment. I'm a little worried about how far you push that analogy, because obviously, as we discussed, artificial neural networks are a very long way from how the brain is operating. And it isn't clear what insights you're getting from this sort of this sort of comparison game. And it's the kind of thing where because we're all worried about sort of the way that AI is running away very, very quickly and exploding in its capabilities, but without any kind of detailed oversight. So a purely nerdy theoretical neuroscience kind of point of view, the idea that you just keep taking this exploding runaway thing and mapping it back to the brain and say, oh, that's how the brain works, isn't a particularly useful way of going either.
I love that answer. That's great. You're the only one that hasn't just immediately said AI in some capacity, like AI automated drones, AI removing our sense of purpose in life. So I like a good theoretical neuroscientist, in-depth, nerdy answer. This is what we're looking for on this podcast. I really appreciate it. This has been such a pleasure. I really do recommend that everyone read The Spike. It's a really well-written book. You don't use technical scientific jargon, which as a communicator I really appreciate. Neuroscience books can be hell with anatomy and just words that don't have any meaning to normal people. Like, you don't say the word. I don't think you have the word action potential in that book, or at least it's very few. It's all spike, blip. You don't use the technical science language. You use very common language. I think that really helps anyone pick it off the shelf and have, massively increased their understanding of how the brain works because of that. I'll link anywhere else people can go find you. Are there any other places where you would like people to come find your work, to come find your writings, to come anything that you're doing at the moment?
So mainly come find me on the Spike at Medium. So that's where I've published a lot of my
Excellent
public essays and my ever popular review of the year in Neuroscience, which is got so popular now that I at Christmas time I get a barrage of texts from other famous neuroscientists going, "Where is it? I haven't seen it."
Amazing. Well, I'll link that down below. I encourage everyone to read it because you are, again, a great writer. Thanks so much. This has been an absolute pleasure. I really appreciate it, Mark.
All right. Thanks, Evan.